Thanks for joining me for another Csec SBA lesson. It's a lab class. We'll be looking at This is lesson 1 of a 2-part lesson. In lesson 2, we'll be looking at how to use the equipment. Without further ado, let's jump right into the lesson. So the objectives for this lesson: at the end of the lesson, students will be able to identify laboratory equipment used for as are outlined in the syllabus, outline the importance of labs, identify types of errors, outline causes of error, identify and use SI units and fundamental quantities. So these are some of the things we're expecting to cover in this lesson. I do hope that you have grabbed yourself a pen, a piece of paper, and at any time throughout this lesson, feel free to stop, rewind, and make your notes. So, list of equipment as are outlined by the Csec syllabus. You are supposed to be able to use and properly manipulate a Bunsen burner with the tripod stand and wire gauze, a binocular and monocular microscope. You are supposed to be able to use both. A measuring cylinder, 250 centimeter cube to 100 centimeter cube. You are supposed to be able to use these carefully and efficiently. A beaker, a 50 cm cube to 500 cm cube. And you're going to be seeing some of these in the videos that will follow this lesson. List of equipment continue. You're supposed to be able to use a thermometer, and we'll be looking at a laboratory thermometer and a clinical thermometer. You're supposed to be able to use a ruler, and a ruler means ruler as small as that ruler that will be in your geometry set to as much as a meter rule. And I do hope that by now everyone would have secured themselves, the persons who are doing science more so, would have secured a geometry set that has a stencil for science equipment. You should be able to use a stopwatch, or you should be able to read a clock. You should be able to read a balance, and in some cases you call it a scale, but we'll look at the different types that we have in the lab and see how best we can help you to manipulate them. You should be able to distinguish between boiling tubes and test tube, and you should be able to use them as they should be used. You should be able to identify and properly use test tubes and test tube holders. You should be able to use a hand lens without endangering yourself or anyone or anything. So all these we will be looking at, not in this lesson, in another lesson, but we'll be looking at pictures or videos of some of these at the end of this tutorial. A syringe, yes, a syringe pretty much like the one that they use in the hospital. You are supposed to be able to use this and use it effectively. Like I said, those equipment are straight from the Csec syllabus, and the syllabus is suggesting that you are supposed to be able to use all these equipment for your labs. Now it's important that we find out, really. Student must be asking from time to time. I'm hearing the grade 9 students asking, "Sir, what's What is a lab? Why do we need to do a lab?" And all these we have tried to cover in the tutorial sessions on labs. Just before school gets started, I think school should be started, get started tomorrow, or the 5th of October, somewhere there. Now the skill, this is a skill. What is Let's look at what is Now is a skill that seeks to ensure that the student can, 1, use different measuring equipment to measure accurately. Use different measurement equipment to measure accurately. Students are also supposed to, they should be able, in all cases, use the equipment to avoid parallax. And parallax is an error in measurement that we will look at later in this tutorial. They should be able to properly execute techniques while using equipment. Select and use the most appropriate equipment for measurement. Record measurement using appropriate SI unit. So a very interesting lineup. You can just look at it. Remember, you can pause your screen at any time and you can write, make notes, go over. It's very important that you understand this content very well so that you'll write a wonderful lab. Now, types of error in measurement. Now, errors can be classified into two groups. It's either the error is going to be systematic or it's going to be random. So the errors that we experience in science, they might be systematic or random. Now, systematic errors. These are errors caused by faulty equipment. Now, every student can relate to this. You have a ruler in your geometry set or in your bag and it fell on the ground and the end of the ruler got damaged and you continue to use it, not paying much attention to the fact that a part of the measurement might have been gone as a result of the ruler being damaged. Now, in some cases, the equipment is not properly calibrated. For whatever reason that happens, I don't know, but in some cases, the equipment is not properly calibrated. Now, the error may also be caused by improper use of the equipment. Now, we spoke about parallax earlier. If you look at the diagram here, A, B, C, it speaks to the position that one should be in when taking a measurement. So we take it that whatever measurement that we're going to take is just at the base of those three arrows. Whatever measurement you are going to take, it is at the base of these three arrows. And when I say at the base, I'm talking about right at this area. Look at your screen. You can see me moving the mouse above parallax. So right at this area, we want to take the measurement. Now, my question to you, where should you be taking this measurement? Should you be standing at A? Should you be standing at B? Or should you be standing at C? Now, if you are standing at A, will the measurement be correct? If you are standing at B, will the measurement be correct? Or if you're standing at C, which will be correct? Now, I want to answer this question for you, but I want you to pull your ruler out of your geometry set or some measuring equipment in the house, anything. And I want you to just hurry on and see if you could try to measure something on the ruler. Have somebody hold the ruler, get yourself into position A, position B, and position C. And I want you to write down all three measurements and compare them. And then I want you to look at the actual measurement and see the degree of error that you would have had as a result of parallax. Now, for this tutorial purpose, or for any measurement purpose or measuring purpose, it's very important you stand directly over or directly in front of whatever equipment you are using to measure. In some cases where you can be at eye level, which you should always try to be at eye level, more so when you are using the measuring cylinders, when you are using the beaker, you want to get it at eye level so you can take a proper reading and you are reading from the meniscus be that you're reading from liquid in a measuring cylinder. So we're going to move on. So here, systematic errors, let me go over it again. These are errors caused by faulty equipment. In some cases, the equipment is not properly calibrated. The error may also be caused by improper use of the equipment. If you're going to be doing a reading, the best place to stand, as it were, for this diagram, it is at position B. Let's move on. The other type of error is random errors. These errors tend to occur irregular. They have an irregular pattern, as we would say random. They are associated with size and sign. Size and sign. Fluctuation in experimental condition is responsible for most of these errors. So for example, you're taking the measurement of some liquid in a measuring cylinder on your decks, and there is somebody who keep moving about and bouncing the decks, and you find that you might be off with your measurement. Of course, you could also have faulty reading. If you have the same person keep taking the reading over and over and over, then this might expose your group to some challenge. You might be experiencing random error. You need to rotate the person that is doing the reading, give them adequate time, ensure that they are seeing well. If they have to be wearing eyeglasses, ensure that they have it on, right? So make sure that you're doing good reading. Now, fluctuation in temperature. So you want to find out about the temperature of water. You are doing some tests with your thermometer, and because the wind keep blowing, the fire on the Bunsen burner keep moving away and almost out sometime, and that type of thing, right? This fluctuation in temperature would affect the heating of the water. The fluctuation in temperature could also affect your transpiration experiment. So all these things, fluctuation in temperature, can definitely affect your reading. When this happens, it is classified as a random error. Now, fluctuation in the wind speed. The wind blowing, the wind speed is just not constant. That too could cause some challenge for the reading. Now we have the list of equipment again. I'm not going to go through them again, at least for now, until we get to the videos, which I'm going to be tying into this lesson at the end. So stay tuned. We want to look, however, at what we refer to as fundamental quantities. Why we want to look at fundamental quantity? It is because in science we use the SI unit, not the imperial unit of measurement. So I know that you are saying, when I went to the shop I bought big gill of oil and 2 pounds of chicken parts and 5 pounds of rice. When you get to your science class and you are doing your measurement, those measurements cannot be used. So the system is based on these quantities that cannot be expressed in any other physical quantity. This is sort of the base. They form the base for what we call derived quantities. Now, because this is a biology tutorial, we are not going to get in depth with this, but I'm sure as soon as we touch the physics, we are going to be understanding a little more about fundamental quantities. The SI unit. You would have covered some amount of this in grade 10, so for most persons this might be very well recall. But if it is not recall, it's a good time to learn. It's a good time to learn. The SI unit are the standard units of measurement defined by the International System of Units, the SI unit, for 7 base quantities. Now we're going to be looking at those now. So let me go over again. The SI units are the standard units of measurement defined by the International System Unit, SI, for 7 base quantities. So the fundamental quantities and SI units. Fundamental quantity. Mass. So we spoke just now about 2 pounds of chicken parts. The fact that we're buying in pound, it is mass. We are not going to talk. Oh. Right, so we're talking about, very good, very good. I thought I made an error just now, I'm very sorry. So mass represented with small m, length represented with a small l or a common L, time, and you can see your screen clearly. Let me hope you are following. Mass there, length there, time there, current here. We're not going to be using current in biology, but the guys who are doing physics, they are going to be using it. Temperature here, and I want you to, this might be a new term for a lot of persons, Kelvin, but I've also done shown the conversion, the formula for conversion at the bottom here. Right, so 0 Kelvin would equal to minus 273 Celsius, and that is, we want to use Kelvin in our science classes. So when you get to class, just let it be a little fancy. Yes, most of the thermometers that you have in your labs, they would have been in degrees Celsius, so you are going to do the calculation to convert it to Kelvin. And you could just ask your teacher to help you, or you could just leave a comment and we will do another video that will help. Alright, so fundamental units, these are the base units, and you are going to be learning later about derived quantities, but we're not going to be looking at that today. Now the SI unit, kilogram. So when we're talking about kilogram, the symbol for kilogram is small k small g. Right? Length is measured in meters. Are you following? You have to look at the screen, you know. So mass is measured in kilogram. Length is measured in meters. Wonderful. Time is measured in seconds. Great. And I'm going to be jumping over current and say temperature is measured in Kelvin. Somebody is saying, but sir, you did not look at the amount of substance. That's more for chemistry. Mole for chemistry. When I do basic chemistry with you, I will talk more about that. And we have luminous intensity. That again, I won't be looking at. Your physics teacher will be looking at that. But this chart is relevant to chemistry, physics, biology. Alright, let's move on. Remember here, it is 0 Kelvin, that equals to minus 273.15 Celsius. We have the list of equipment going again. Alright, so it's time now to hop on over to the videos. The use of each equipment will be demonstrated in separate videos. Alright, you have to watch the videos carefully. For example, when you see the video that has the Bunsen burner, the tripod stand, and the wire gauze, they'll be covered in one video. I'll try my best to take you through it.